So, good morning or good afternoon to everybody. It's 9 a.m. at the West Coast and 6 p.m. in Europe. It's my pleasure to continue our Global Immunotalk series by introducing another excellent speaker, Daniela Thommen. And it's really a pleasure to have you here today. I'd like to say a few words about you. So you are a group leader at the Netherlands Cancer Institute right now. You started your career with receiving an MD PhD degree, which you obtained from Switzerland, in Basel in Switzerland, and this was in 2010. Afterwards, you started both a clinical career and a research career. So you were for a short period a resident in internal medicine at the hospital in Basel. You then joined the group of Alfred Zepelius, where you did research, where a postdoc, and in 2016 you returned. I think you returned. So I think you're from the Netherlands. You have to correct me if this information. Where are you from? Correct me, please.
I'm actually
I can't
Swiss.
hear you. You are Swiss. Oh gosh. Okay, I'll put you into Netherlands. But okay, good. Oh, so then you moved forward to the Netherlands in 2016. You joined the lab of Ton Schumacher at the Cancer Institute in Amsterdam, again as a postdoc. In 2020, you started your own lab as a junior group leader, obtained tenure in 2023. And in 2024, you joined the OnCode Institute as a junior investigator. You're doing spectacular research, as we will hear in a minute. And but quickly summarizing, your research focuses on understanding the role of intratumoral immune cell heterogeneity and consequences that result from this heterogeneity on immunotherapy responses. You're very interested in the development of personalized immunotherapies and strategies, and you use different types of organotypic models from humans to address those questions. You won numerous prizes. I'd like to highlight a few. So in 2018, you got the Bas Mulder Award from the Dutch Cancer Society. In 2019, the Swiss Pfizer Research Prize in Oncology. 2022, the NWO VD Award, and in 2023, the Aspasia Award. And I could mention other things. I could talk about fellowships and funding and so on, but I don't want to waste too much more time because we want to hear your research. And so I will finish my introduction with asking you the typical question, and here what we have discussed what I want to discuss to ask you was if you could share with us the most impactful decisions in your scientific career.
Yes, so thank you, Dietmar, for the very kind introduction, and yeah, thank you for the question. And I think it actually relates very much to my CV. So I think for me, probably I made many questions or many decisions that had some impact on my scientific career. But I think the most impactful was the decision to go abroad to the Netherlands for a postdoc. Because as you said, I trained as a MD. I did my clinical specialization in medical oncology, and I never imagined I would not stay in the clinic and work as a doctor. And at one point I decided after doing my PhD and my first postdoc that I really want to spend some time on science and doing research in the lab outside of Switzerland. So I moved to NKI, to the lab of Ton, and encountered an entire new world of research that was very inspiring, very collaborative, really different approaches to science that intrigued me so much that, despite having already a position secured as an attending physician, I decided to stay in the Netherlands, so not go back to Switzerland, and also to leave the clinic and not seeing any patients anymore, and becoming a group leader and having a research lab, something I never could have imagined when I left for my postdoc. So I think what I learned from that is it's good to have plans, but it's even more important just to be open for new challenges, new opportunities that may come up, and really change not only your career but also your life a little bit. Yeah.
Well, it's very interesting to set up because I I essentially did the same at some point. But my question is, do you miss the patients?
Well, I do miss the patients. I mean, I really liked the clinical work. I do not miss the administrative part. So, and I hope in the end that with my research I can create more impact for the patients, or more better cancer treatments than I can while treating the patients myself. Yeah.
Okay, we had a small interruption. I'm not quite sure if the interruption was on my side or on your side, but never mind. We will just continue from here. So it's a pleasure to have you. And now it's all up to you. So please share your slides, and we are keen on hearing your presentation.
Thank you. Okay, I will try to set up my slides. So I hope you can see them in full screen now.
Looks all
Okay,
good.
so great. All right, then first I would like to thank again the organizers for the opportunity to participate in this Global Immunotalk series. And what I would like to do today is to share some of our work over the past years in which we try to dissect the heterogeneity in T-cell activity in human cancers and to understand how we can leverage this for cancer therapy. So when T cells recognize their cognate antigen, for instance, in acute infections, they differentiate into effector cells and acquire functions such as proliferation and secretion of cytokines or cytotoxic mediators that then lead to the elimination of the infected or foreign cells. And after the antigen is cleared, these cells transform to memory states that then can mediate the long-term protection. However, in other settings, such as, for instance, chronic infection or cancer, this process doesn't work. The T cells cannot clear the antigen and therefore are exposed to chronic antigen simulation, which induces a state called dysfunction or exhaustion. And the state is characterized by overexpression of so-called immune checkpoints and a gradual loss of effector functions. And one major key inhibitory receptor in this process is PD-1. So PD-1, PD-L1 signaling has really been established as a major driver of this dysfunctional state. And it has also been shown that blockade of this pathway can reinvigorate T cells and restore effector functionality. And this treatment is now quite widely used in the clinic. So here you can see a plot with the response rate in different cancer types. And you can see that PD-1 or PD-L1 blockade shows activity in all of these cancer types. However, the response rates vary quite a lot. And what you can also see is the majority of the tumors still have very low response rates, or usually around 15 to 20%, meaning that there are still many patients that currently do not benefit from these treatments. So we are still struggling to understand why patients respond to this treatment or not, and how we can treat patients that are resistant. So therefore, in the lab, we are interested to understand the mechanisms by which PD-1 blockade remodels the immune response in human cancer tissues, and also to identify barriers that potentially can limit T cell function in tumors that are resistant to PD-1 blockade. So to address these questions, we first need to understand basically the T cell landscape in human cancers. And when we started to look into these questions, that was quite long before single cell RNA sequencing technologies were available. So we used an approach to investigate this that was inspired by data from the Very lab at this time in chronic LCMV infections, where they found that PD-1 positive virus-specific T cells can be divided into two subpopulations defined by either intermediate PD-1 expression or high PD-1 expression level. And what was quite interesting is that the PD-1 intermediate cells were more early dysfunctional, so they still had a high proliferative capacity, and they could be reinvigorated by PD-1 axis blockade. In contrast, the PD-1 high expressing cells were more late dysfunctional, or what we view classically as exhausted. They could not proliferate anymore, and they did not respond to PD-L1 or PD-1 blockade. So we found this quite interesting and wanted to understand whether the same is also happening in human cancers. And for that, we looked at populations with either negative, intermediate, or high PD-1 expression. And then we asked the questions whether we can also find these PD-1 high and PD-1 intermediate T cells, and whether they also would represent more early and late dysfunctional cells. Which of these subsets actually has the capacity to recognize cancer cells and to be tumor reactive, and also whether we can find differences in these subsets between responding and non-responding patients upon checkpoint blockade therapy. So to do this, we first needed to establish a reasonable cutoff for identifying these subsets. And we did this by using a reference-based threshold from peripheral blood. So you can see here a flow plot showing PD-1 expression in CD8 T cells in blood. And these cells usually express PD-1 at a more intermediate level. Whereas in the tumor, we find sometimes CD8 T cells that express this very high PD-1 expression levels that we actually don't see in blood. So we term these cells as PD-1T for this tumor-specific PD-1 expression and compare them to PD-1 normal, that are the intermediate cells that have the same level as those in the blood, and PD-1 negative T cells. So we first sorted these cells from human lung cancer samples and then cultured them ex vivo with stimulation by anti-CD3, anti-CD28 antibodies to look at the production of cytokines. And what we found is that, as expected, the PD-1 T cells, so the high expressing T cells, did not produce any effector cytokines like IL-2, TNF-alpha, or interferon gamma anymore, even when they were stimulated, in contrast to cells with a more intermediate or low PD-1 expression. So suggesting that these PD-1 T cells are really dysfunctional. So to further characterize them, we also performed bulk RNA sequencing analysis of the sorted subsets. And there we were already a bit surprised to see that when we look here at the principal component analysis, that this PD-1 T cells here indicated in red seem to be very distinct from cells with intermediate or negative PD-1 expression. And we also can see this when we look at the number of significantly different genes. So the high expressing cells are really different from both the intermediate and the negative cells, but the latter two populations actually are very similar and hardly display any differentially expressed genes. So that was a bit surprising because we expected the PD-1 highs to be more similar to the intermediates than actually the other two populations. When we then look at selected genes in these subsets, we do see, again, a lot of genes associated with dysfunction in the PD-1 high expressing cells, like a couple of immune checkpoints, PD-1, TIM3, CTLA4, TIGIT, or LAG3, so all in line with these cells being a dysfunctional population. However, what we also found is that these cells acquired expression of new effector molecules, like CXCL13. So this is a chemoattractant for B cells that is normally not produced by CD8 T cells. So therefore, we first wanted to understand whether these cells actually can secrete CXCL13. And we tested this both in the supernatant of sorted subsets and also by intracellular staining. And we could confirm that these cells actually also at the protein level can produce CXCL13. And more interestingly, if we look at the number of cytokines and chemokines, this seems to be more or less the only one produced by this subset. So first of all, this indicates that these dysfunctional PD-1 T cells are actually not functionless, as is often implied when thinking of exhausted cells, but that they acquire an altered or perhaps more adapted function in the tumor microenvironment. And then, of course, the question is, why do they start to produce CXCL13? So CXCL13 is a chemokine that is crucial for the formation and maintenance of lymphoid tissue. And lymphoid tissue can occur in tumors in the form of so-called tertiary lymphoid structures, or TLS. So these are lymphoid aggregates dominated by B cells and T cells that can form in chronic inflammatory settings, such as tumors. And they are thought to promote immune cell recruitment and also perhaps being a site for local immune cell activation in the tumor. And more importantly, they also have been recently associated with response to immunotherapy in a number of cancer types. So therefore, we wondered, could there be a link between this dysfunctional PD-1 T cells and the TLS? And for that, we've next used immunohistochemistry and immunofluorescence analysis to quantify PD-1 T cells inside and outside of TLS. And what we found is that these cells actually really predominantly localize within TLS and are hardly found outside in the tumor. So suggesting that indeed there might be some connection between the cells that start to produce CXCL13 and the formation of this TLS. And this is something that we're still very interested in and now working on to further understand how this really connects and how these structures might contribute to immunotherapy response. However, as a next question, we actually wanted to understand whether these cells are important. So first of all, do they actually have the capacity to recognize the tumor cells? And for that, we again sorted these different populations from lung cancer samples, and then we expanded them over 2 weeks in high-dose IL-2 to generate the necessary numbers for functional testing, and also to reactivate them, that we have like a similar basis to compare. And then we co-cultured them with autologous digest. And here you can see the interferon gamma production in these subsets upon co-culture. And what is already visible is that interferon gamma production seems to be clearly higher in the PD-1 T cells than in the other subset. And that's actually something we observed very consistently across samples. So in all tumors that we tested, dysfunctional PD-1 T cells were able to recognize the matched tumor digest, whereas this was only the case in very limited numbers for the cells with intermediate or negative PD-1 expression. So altogether, this was quite interesting. So on the one hand, these cells seem to be quite dysfunctional, but they also enrich for tumor activity. So we wondered how they would correlate with anti-PD-1 response in patients. And to test that, we quantified these cells in a small cohort of 21 patients with advanced lung cancers that were either responders or non-responders to PD-1 blockade. And what we found is that was actually quite a striking difference, that these PD-1 T cells were clearly enriched in tumors that showed a clinical response to PD-1 blockade compared to non-responders. And also, we found that the correlation with response was clearly superior to PD-L1 expression, which is the currently used biomarker to stratify patients. So based on this observation, we wondered, could we use the presence of these cells as a biomarker in the clinic to select patients for this treatment? And this was a study that we finished recently that was led by Carlijn Hummelink, PhD student, in collaboration with Kim Monkhorst and Greet Meijer from pathology at NKI, and two pathologists from Switzerland, Victor Kölzer and Kirsten Mertz, where we first actually had to develop technology to quantify these cells in FFP pretreatment tissue biopsies, because beforehand we characterized the cells by flow cytometry, and that's of course something that is not really useful in the clinic or to do this in large clinical cohorts. So what we had to do is basically to translate this tumor-specific PD-1 expression level into an IHC assay where we can capture these cells, because it really doesn't work if we just look at PD-1 positive cells. So therefore we used an algorithm-based approach, establishing a digital image analysis protocol that allows us really to capture cells with a specific intensity of PD-1 expression in the tissue. You can see here that if we do this for matched samples, in the end, we have a pretty good correlation between the PD-1 T fraction by IHC and the one by flow. So we used this approach to quantify PD-1 T cells in pretreatment biopsies of 120 patients with advanced lung cancer that were undergoing PD-1 blockade treatment. And similar to a smaller cohort before, we observed that patients with disease control at 12 months, so that did not show any tumor progression upon treatment, had high levels of these cells in their tumors compared to non-responding patients. And even more important, we saw that patients that have very low or even no PD-1 T cells in their tumors have a very poor chance of responding to PD-1 blockade, so suggesting that these patients should not receive immunotherapy alone. We also saw that higher infiltration by PD-1 T cells in the tumors was associated with a better overall survival compared to patients with low PD-1 T cell fractions. And when we look at the sensitivity of the biomarker, this is actually pretty good, with more than 90%, and clearly superior to one of PD-L1 in the same cohort, that we tested at 2 cutoffs that are used in the clinic, and that clearly performs less well. So we recently finished developing a clinical grade assay that allows us to do this in a diagnostic setting, and we hope that we can soon test this prospectively in patients and then really move forward as a clinical test. So to conclude this first part, we found that these PD-1 high or PD-1 T TILs in human cancers really form a distinct dysfunctional subset that is highly enriched for tumor reactivity. And importantly, that these cells are not functionless, so they clearly lose their classical effector functions, but also acquire new functions by CXCL13 production. And also that the presence of these cells seems to be predictive for clinical outcome to PD-1 blockade. So this suggests that these cells are correlated with immunotherapy response. However, we still don't know whether these cells actually still can respond to PD-1 blockade. And to test that, we need model systems that allow us to really look at the early reinvigoration of T-cell immunity upon immune checkpoint blockade. And there are different ways how people have done that. Of course, by using animal models, which were really critical in, of course, discovering the immune checkpoints and also to gain some mechanistic insights. However, we also know that sometimes it's really challenging to capture the heterogeneity that we see in human cancer patients. Another way is to look at serial clinical biopsies that, of course, capture the whole spectrum of heterogeneity that we see in patients, but we cannot really do mechanistic experiments, and we also often just miss really the early response as these samples are taken weeks after treatment start. So we want to bridge these two approaches by developing and using ex vivo model systems that are really based on patient-derived material, but that they still offer the possibility to perturb cell function, so to do mechanistic studies, and where we can also directly compare multiple interventions side by side in the same tumor. And one such model that we developed is what we call the patient-derived tumor fragment or PDTF platform. So for that, we collect material from patients undergoing surgeries, and then we dissect the tumor tissue into small tumor fragments or PDTFs. We do this for 10 different cancer types at the moment. So really from highly immunogenic to lowly immunogenic cancers. And then we can culture these fragments ex vivo, treat them with immunotherapies, and combine this with many different readouts to really investigate what the treatment does in the tissue. We can also cryopreserve these fragments, and we have so far established a tissue biobank with viable material of more than 600 patients. So this is a picture how these cultures look like. So you can see here these tiny fragments in the 96 well plate. These fragments are about 1 cubic millimeter in size, and they really preserve the composition of the tumor microenvironment, the architecture of the tumor, but also the activity of the immune cells in the tissue. So we use this platform in a first study to basically see how tumors immunologically respond to PD-1 blockade. And this was a study done by Paula Worbil, Marlijn de Bruin, and Lisanne Roelofsen in the lab, where we cultured fragments from 37 tumors from 5 different cancer types, either in the absence or presence of PD-1 blockade. We always culture multiple fragments per treatment to capture potential heterogeneity within tumors. And we do this really for short term, so we can already after 48 hours see the effects. And we looked here at a number of T-cell activation markers and cytokines and chemokines. So this heat map shows the changes that are induced with PD-1 blockade in the fragments. On the top, you can see the different tumors. On the right side, the parameters we measured. And the color here indicates the change with anti-PD-1. So the more red means the more change. And what we found is that there seem to be 2 groups of tumors. One to the left, where basically nothing is happening with the treatment, and the tumors to the right, where we see an increase in quite many of these parameters. So based on this immunological response, we defined these tumors as either PDTF non-responders or PDTF responders. So next we wondered how relevant these responses are. So basically, would they also correlate with the clinical response of the patients? And to do that, we first developed a response score. So for that, we identified the 12 most discriminative parameters between responding and non-responding fragments. And then we made a score out of that. So you can see that here plotted in this cohort that I just showed you, with the responding tumors in purple and non-responding in gray. And then we can directly compare this with the clinical response of the patients. And in this cohort, 12 patients also received systemic anti-PD-1 treatment in the clinic. And you can see that for these patients, we here have a perfect match between the fragment response and the clinical response. So we went on then to validate this in a second independent cohort. And also here we found that, with the exception of these 3 patients indicated here in blue, that clinically had a mixed response, and where we don't know whether we observed a responsive or resistant lesions, we again see this strong correlation between fragment response and clinical response, so suggesting that this early immune activation in the tissue is highly predictive for clinical outcome. So this now offers us the opportunity to use this model to really take a closer look at these immunological responses. And as the first question, we ask, can we now see activation of T cells by PD-1 blockade? And for that, we first used flow cytometry. So we looked at the number of T cell activation markers that you can see here. We see on T cells in unstimulated fragments that these markers are very lowly expressed. And in this tumor with PD-1 blockade, we see that these markers increase. And in parallel, we could also see similar effects on the induction of effector cytokines like IL-2 or interferon gamma, and the secretion of cytotoxic mediators, so suggesting that indeed this intratumoral T cells can get activated. So we next wondered how much they contribute to this broader immunological response that we observed before. So the heat map I showed you with all the cytokines and chemokines. And for that, we wanted to block T cell activation in the fragments. And to do this, we used an LCK inhibitor. So LCK is downstream of the TCR, meaning if we block here, the TCR signal will be stalled and the T cells will not get activated. So we compared tumors that we treated with either PD-1 blockade alone or with LCK inhibitor pretreatment. And you can see here whether all these tumors show a nice response with PD-1 blockade. This response is largely gone when we block T cell activation, so suggesting that these intratumoral T cells are really key drivers of this local anti-PD-1 response. So this was quite interesting, but of course also just very superficial, because we just looked at the total T cells. So next we wanted to understand which specific T cells now get activated by this treatment. And of course, we are particularly interested in the dysfunctional T cells, but I already showed you before that these cells can have differences in PD-1 expression. And of course, now with single cell technologies, we can look at this in much higher resolution. And I think what we have learned over the last years is that dysfunction is not really a uniform state, but also more a gradient of cell states, ranging from more precursor-like cells to transitional to more late dysfunctional cells. And especially these precursor-like cells have been very well studied. It has been shown that these cells have more stem-like capacities, they're highly proliferative, often reside in the lymphoid tissue, and they're characterized by expression of the transcription factor TCF7 and express low to intermediate levels of PD-1 and other checkpoints. And what was particularly interesting to see is that in mouse models, these cells really seem to be essential for durable response. On the other side of the spectrum are the more late dysfunctional cells. So these cells are more residing in the tissue, and they are the cells that are classically viewed as exhausted. They express high levels of TOX, which is the transcription factor that is really driving this exhaustion program in the cells, and they express high levels of PD-1 and other inhibitory molecules. And it is very unclear whether these cells still have any capacity for reinvigoration or whether they are just too far gone to do anything. However, we find these cells quite interesting because, of course, I showed you they correlate with immunotherapy response. We know they are highly tumor reactive. They also express effector genes, acquire new functions. So we thought perhaps they still do something. So to investigate that, we started a study led by Pauline Kaptein and Nadine Slingerland, together with the group of Ton Schumacher that is still very much ongoing work. So I just show you some first preliminary data. So what we did is we combined the PDTF cultures with single cell sequencing. So we did that for 5 tumors treated with or without PD-1 blockade. 3 of them were responding to the treatment, 2 of them non-responding. And I show you here the UMA plot of the CD8 T cells. So each dot is a cell and all the colors indicate different CD8 T cell states. And what we could see is, so we can use gene expression signatures to annotate these cell states. And what we could see is there seem to be 3 dysfunctional states. We have one here in orange that is a bit less dysfunctional. You can also see low expression of these exhaustion signature, whereas the other 2 seem to be more late dysfunctional. And interestingly, this green state here also seemed to express some effector genes. So to further understand how these cell states are related, we looked at TCR sharing, and we found that all of these 3 dysfunctional states share TCRs, but that especially these 2 late dysfunctional states show quite high TCR overlap. So we wondered whether these states actually could be reflective of different treatment effects. And to test this, we quantified the cell state fractions in the different conditions, so in unstimulated fragments and in anti-PD-1 treated fragments, and we did this separately for non-responding and responding tumors. And what we found is that indeed this green dysfunctional state seems to come up specifically in the responding tumors with PD-1 blockade, so suggesting that these cells really expand upon treatment. And when we further characterize them, we also see that they express still many dysfunction genes, but also they upregulate many genes related to T cell activation, and they enrich for biological pathways such as T cell receptor signaling, regulation of immune response, T cell differentiation, cytokine production, or cytotoxicity. So suggesting that this is really a population that gets reinvigorated at the tumor site, and that seems to mostly come from actually the late dysfunctional cells. So, of course, that's very interesting, but then we asked, okay, is this something that we actually just see in the fragments, or is this something that is really relevant in patients? And for that, we were very fortunate to collaborate with Lotje Sur, who is a head and neck surgeon at NKI, and who performed the Matisse trial. So this is a trial in which cutaneous squamous cell carcinoma patients are treated with 2 cycles of immune checkpoint blockade before curative surgery. And this works quite well. So here you can see an example of a patient that had these large tumors before treatment, and then after only 2 doses, these tumors were really like melting away. And we see that there is a really high number of patients that have a major pathological response. So this is very, very exciting. What we also did is we designed a quite extensive translational research program for this trial that is led by Mercedes Machuca-Ostos, who is a PhD student in both our groups. So what we did is we take a lot of serial biopsies from these patients, both for tumor and blood. And we do this at baseline in week 1, week 2, and week 4. And importantly, we also stratify the week 1 time point from as early as day 2 to day 7 to really capture the very early dynamics of the T cell response. So we then performed single cell RNA sequencing of these samples, and we also established PDTFs that we treated then with the same treatment as the patients, and where we also performed single cell sequencing, so allowing us to really uncouple the local and systemic effects of the treatment. And also this is still very much work in progress, so I will just show you one piece of data. Here again, we look at the CD8 T cell states from the biopsies. you can see here, again, the dysfunctional compartment with different dysfunctional state and also in green proliferating states. And we can then see or follow the changes in these states over time with the different time points of the biopsies. And here, this is shown as density plots on the UMAP. And what you can see is that we seem to have very early in week 1, mostly an increase in this proliferating and dysfunctional states, also week 2, and then in week 4, a transition towards more memory or resident memory states. And we now can compare this to what's happening in the fragments in exactly the same patients. So here I just show you the different T-cell states and a T-cell activation score. And we found that this also specifically increases in the dysfunctional and especially the proliferating states, so really mirroring the early changes in the patients. So concluding from this, we see that PDTFs are actually a quite reliable model to recapitulate immunological responses, and especially T-cell reactivation upon checkpoint blockade that is also observed in cancer patients. And we see that this early immune activation in the tissue is highly predictive for clinical response. And interestingly, we also see that, at least at the tumor site, dysfunctional T cells seem to be the key drivers of this immune reactivation. So we think that PD-1 blockade, or a model how PD-1 blockade works, could be that it leads initially to this local reinvigoration of the more late dysfunctional cells. And we know from work by other groups that this is probably not a durable reinvigoration, as the cells might be just epigenetically in a fixed state and do not have much capacity for proper rewiring. But it probably is enough to really induce this pro-inflammatory local response, and especially a secretion of chemoattractants that then can mediate the recruitment of probably less dysfunctional T cells from the lymph nodes to the tumor site that then can take over the response. And this is also something that we are now trying to address in the Matisse trial. So for the final part of my talk, I would like to switch gears a bit from anti-PD-1 responsive to anti-PD-1 resistant tumors. And of course, there the big question is, why are these tumors resistant, and what can we do to treat these patients? And one interesting observation we made when we analyzed tumors that were non-responsive to PD-1 blockade is that actually quite a large fraction of these PD-1 non-responding tumors still is infiltrated by T cells, actually quite comparable to the responding tumors. So this suggests that there might be barriers other than PD-1, PD-L1 signaling that can limit the functionality of these T cells. So we started to profile these T cells, and one observation we made was that in these tumors, there's often very low levels or even lack of interleukin-2. So IL-2 is a cytokine that is very critical for T cell activation and effector differentiation. And it has also been found to be required for response to PD-1 blockade. So these are data from mouse models where you can see that with PD-1 blockade, there is tumor control. However, when IL-2 signaling is blocked, this response is lost. So IL-2 is not new. So it was actually the first immunotherapy showing activity in melanoma and renal cell carcinoma long before immune checkpoint blockade. This is an example of an older study from a long-term analysis from 270 melanoma patients that were treated with high-dose IL-2. So the overall response rate was 16%, which is in the range of CTLA-4 blockade. But what was quite interesting is that about 60% of patients that achieved a complete response upon IL-2, they also achieved a durable response. So you can see here this survival plateau, meaning that these patients really reached long-term immunity. So this suggests that IL-2 can be quite potent in anti-tumor activity. However, it is also quite problematic because it is very toxic. So its clinical use has really been challenging. because of that. And as a second problem, IL-2 is also a highly pleiotropic cytokine that can not only lead to CD8 activation, but also to immunosuppressive effects. So, for instance, in an experiment where we tested tumors in ex vivo with either checkpoint blockade or checkpoint blockade plus IL-2, we saw that it really can increase CD8 activations even in tumors that do not respond to checkpoint blockade alone, but it also highly activates regulatory T cells as they express the high sensitivity IL-2 receptor. So we wondered, is there a way to make IL-2 more specific or also to make it less toxic? And for that, we collaborated with a company called Asher Bio, who is developing so-called cis-targeted cytokine therapies. So what they did is they took IL-2 and they attenuated it, so they reduced the binding to the IL-2 receptor alpha, and they also fused it to a CD8 antibody, meaning that this molecule is then specifically delivered to CD8 T cells. So keeping the anti-tumor activity, but losing the effect on other cells that can mediate immunosuppression or toxicity. And you can see here in vitro assay that this works really well. So if we look at recombinant IL-2, we see that this, even at low doses, highly activates Tregs, and that we need quite high doses to also get CD8 activation. And in contrast, the CD8-IL2 really specifically activates the CD8 T cells, and only at very high concentrations also show some effect on the Tregs. So they also tested this molecule in vivo, and these are data from a T3 sarcoma model, where you can see that the high dose of the CD8-IL2 really led to a potent tumor control in the majority of the mice, whereas the untargeted IL-2 was not effective. And importantly, the untargeted IL-2 really showed high toxicity. So you can see here this body weight loss in the mice, whereas this did not happen with the CD8 targeted IL-2. So Pauline Kaptein in my group became interested to test this molecule in the PDTS. And for that, we took again a cohort of 5 cancer types, 23 tumors that at this time were pre-selected on having T-cell infiltration, as we reasoned, that we will not see much in immune desert tumors. And we first, again, looked at the production of cytokine and chemokines, as we found this quite predictive in the PD-1 blockade setting. And similarly, we found again 2 groups. So one group where we do see quite a lot of immune activity upon CD8-IL2, and one group where not much is happening. So we next asked whether this actually correlates with the activation of CD8 T cells. And for that, we looked at a number of different markers. So I show you here granzyme B as a cytotoxic molecule, and CD137 as an activation marker. And here we were quite surprised to see quite distinct effects. So for CD137, this really performed as expected, that it increased upon treatment in the tumors that also showed an immunological response, whereas granzyme B seemed to increase very independent on whether these tumors also start to produce cytokines or not. So as CD137 is downstream of TCR triggering, we wondered whether this could be a sign of a more TCR-dependent effect, and granzyme B perhaps of a more TCR-independent activation. So to test this, we again used the LCK inhibitor setting that we then compared to CD8-IL2 alone. And we indeed found that when we pre-treat the tumors with the LCK inhibitor before adding the CD8-IL2, that we do not see this induction for CD137, but we still do see the effect on granzyme B. And importantly, we also see that the cytokine response only happens when the T cells get also a TCR signal, and especially also the secretion of granzyme B, so suggesting that the cells start to intracellularly produce it, but do not secrete it. So together, this suggests a model that IL-2 can have both TCR independent and dependent effects, with independent effects inducing effector capacity in the cells, but not really effector function. That only is induced when there is also simultaneous antigen recognition. So to further test this, we again performed single cell sequencing of PDTFs treated with CD8L2. And you can see here the UMAP plot with the CD8 T cell states from 5 different tumors. We again see different cell states, the more naive cells on the top, some more memory states, effector states, and the more dysfunctional states. We can also look at canonical marker genes to compare between these different subsets. And what we found is that there seem to be 2 memory states that overall look quite similar, but one of them seems to be a little bit more activated, so having a bit more cytotoxic markers. And similarly, we also see 2 dysfunctional states, of which also one is more activated than the other. And then we also see 3 proliferating states, of which 2, the cycling 1 and 3 states, actually seem to be more similar to the dysfunctional clusters, and the cycling 2 more to the memory clusters. So we wondered, again, could this be a treatment-induced effect? So for that, we plotted again the cell state densities for cells from untreated or CD8L2 treated fragments. And you can see here this profound rewiring of the CD8 T cell landscape with CD8L2, so where basically this whole right part of the UMAP plot comes up with the treatment and is not present in untreated fragments. So when we quantify this, we see that there are 5 cell states that seem to be induced, or at least strongly expanded by CD8L2, that all localized in this right part of the UMAP. And in parallel, we also see that there are 2 states that are mostly present in unstimulated fragments and then decrease with treatment, so suggesting that they perhaps transition towards this more activated states. We also confirmed that these states really undergo effector differentiation. So here, for instance, we plotted gene signatures from effector T cells in acute infections, and you can see that all these IL-2-induced states actually show increased expression of this signature. So to further understand how the cell states are related, we again looked at TCR sharing, and we could see that we had sharing as expected between the 2 memory states, but also between the dysfunctional and the related proliferating states. But interestingly, we did not see much sharing of the cycling 2 state with any other state. So we performed label transfer to better understand from which cells these cells may originate. And we see that they are quite diverse, so coming from many different clusters like naive cells, transitional cells, or memory cells. When we look at the clonal expansion, we see that this is in line with our previous results, where we see this mostly in dysfunctional states. That, of course, we also showed before that they are enriched in tumor activity, also in line with more recent data from other groups, and a little bit in the effector states. However, when we really functionally test them, again by sorting and co-culturing them, we do see that tumor activity really seems to be largely restricted to the dysfunctional cells. So we next wondered which of these activated states now really depend on antigen recognition or not, keeping these differences in tumor activity in mind. And for that, we again used our LCK inhibitor approach. So here I show you a third condition where we also have the CD8L2 pre-treated with the LCK inhibitor. And you can see that this is different from when we just add the CD8L2, but we still do see much more effects compared to the untreated fragments. So when we look at these 5 induced cell states, we also see different dependence on TCR stimulation there. So we have 2 states, so this activated memory and the cycling 2 states that seem to be largely independent of TCR stimulation, whereas the activated dysfunctional and the dysfunctional proliferating states seem to be at least partially dependent. So to further untangle the effects of TCR triggering and IL-2 signaling, we developed 2 gene programs for the respective signals. So for IL-2, we used the fragments treated with CD8-IL2 plus the LCK inhibitor and looked at differential genes to the untreated fragments, reasoning that here we remove the TCR signal, so we should only have the IL-2 effect. And vice versa for the TCR program, we used a condition where we treated with anti-CD3 that was not included in the clustering, and we reasoned that anti-CD3 should deliver the TCR signal to all the CD8 T cells in the fragments, so we can use this to establish the TCR program. So when we look at the differentially expressed genes in this program, we found in the IL-2 program mostly genes associated with effector differentiation, and in the TCR program, genes with T cell activation. When we now plot these programs on the different cells, you can see quite profound differences in expression. So the IL-2 program really seems to be quite widely expressed in all these cells that are, or cell states that are induced by the treatment, whereas the TCR program is much more restricted, mainly to the dysfunctional cells that also show clonal expansion. And interestingly, it doesn't seem to really come up in these effector cells, so further highlighting that they might not recognize tumor antigens. So altogether, this suggests that IL-2 really may induce, broadly induce T cell effector differentiation across multiple cell states, but that the full activation seems to be more dependent on a simultaneous TCR signal. And one additional observation that we made that was quite interesting to see is in our co-cultures, where we sorted the subsets and then co-cultured them with autologous tumor digest. We also included a condition where we added CD8-IL2. And what we observed there is that in this setting, we see an increase in CD137 expression on the T cells, but we do not see any effects on, for instance, degranulation or cytokine production. And of course, we have to keep in mind that before this co-culture, we already expand these cells in high-dose IL-2, suggesting that probably these cells reach already their limit in functional capacity, but they still can increase CD137. So perhaps reflecting that IL-2 may also enhance just TCR signaling strength. And in line with this, we also see, for instance, in fragments treated with anti-CD3, that the addition of CD8-IL2 to anti-CD3 can still further enhance the TCR program score. So suggesting indeed that there might also be an additional effect on TCR signaling. So altogether, we think that IL-2 can reactivate intratumoral T cell immunity by 2 mechanisms. First, by this broad arming of intratumoral CD8 T cells with effector capacity, and simultaneously probably also on enhancing the TCR signal, and that both together enable the dysfunctional CD8 T cell pool in the context of antigen recognition to become reinvigorated. So for the final few minutes of my talk, I would like now to focus on the question we were actually interested in, namely whether this is also something that works in anti-PD-1 resistant tumors. And to test that, we compared PD-1 blockade and CD8-IL2 and the combination side by side. So we took, again, a cohort of a few different tumor types and treated them with anti-PD-1. And you can see here 4 tumors already respond to this treatment, whereas the rest is resistant. And then we can compare this to the same tumors treated with either CD8-IL2 or the combination. And you can already see that we increase the number of responding samples. So when we look at matched samples, we see that actually 44% of the tumors that are resistant to PD-1 blockade can show immunological activation when we treat them with either CD8-IL2 alone or together with anti-PD-1. And to really understand how this treatment acts on the T cells, we used tumors again included in our single cell cohort. So 3 of the 5 tumors were both responsive to CD8-IL2 and to anti-PD-1. So we can directly compare the signals or the effects in the same T cells. So here I show you again the density plots from the different treatment conditions. And I think what is really striking is the profound differences in transcriptional rewiring between CD8-IL2 and anti-PD-1, despite quite similar cytokine production that we can pick up in these tumors. So to have a more fair comparison, because we know now that IL-2 also has some TCR-independent effect that we would not expect with PD-1 blockade, we really wanted to compare the effect in tumor-reactive cells. And for that, we inferred tumor reactivity using 2 recently published transcriptional signatures that have been functionally validated in neoantigen-specific T cells. And we bind our cells into tumor activity high, intermediate, or low. And when we first look at tumor activity high cells for the different conditions, we found, as expected, that in untreated fragments, these cells are largely dysfunctional, as you can see here, indicated by this red state. And we see that with anti-PD-1, actually a fraction of these cells acquires this more activated dysfunctional state, but cannot really transition into this highly activated proliferating states. And this is quite in contrast to CD8-IL2, where almost all of these cells are reinvigorated and really also acquire these highly proliferating states. And more interestingly, also, when we look at cells with intermediate tumor reactivity, we still see an effect of CD8-IL2, that at least a fraction of these cells is getting activated, which clearly does not happen with PD-1 blockade, so suggesting that there is perhaps even a broader repertoire of cells that can be mobilized by this treatment. And as a final observation, we also looked at TOX expression, because this is the transcription factor that is really driving this functional program. And you can see here that CD8-IL2 really substantially reduces TOX expression in these cells, so suggesting that perhaps these cells may even undergo a more durable reinvigoration than with PD-1 blockade. And this is something that we're also still very interested to explore. So to conclude this final part, we found that IL-2 can, or lack of IL-2 can present an additional barrier that can limit T cell function, and that targeting of IL-2 to CD8 can really specifically reinvigorate CD8 T cell immunity, probably by arming the T cells with effector capacity and then allowing them to really acquire effector function when they get also TCR-dependent stimulation. We think that CD8-IL2 actually can induce a broader and qualitatively superior activation of tumor-specific T cells than PD-1 blockade. And we see that it can also work in anti-PD-1 resistant tumors, so suggesting that at least some of these tumors may harbor a tumor-reactive T cell pool that is not responsive to PD-1 blockade alone. And just as a side note, this molecule CD8-IL2, also called AB248, is currently in phase 1 clinical trials. So we are very curious to see how this will do in patients. And with this, I would like to really thank all the many people involved in this work, especially my fantastic lab who did all the experiments and all the work underlying the data I showed you today, our collaborators at NKI and outside NKI at Ashur Bio, NKI facilities that really support us a lot with their help, our fundings, and thank you for your attention.
Well, thank you very much, Daniela, for this great talk and for sharing the excellent data with us. As usual, it's now time for the discussion, and I know we would all like to discuss this, and I also have many questions, but as before, I have to refer for that to our Twitter account. You see the connection, so please ask as many questions as you like to Daniela, but use it through Twitter. With this, we're at the end. Thank you very much again, everybody, for joining us for your presentation, Daniela. And I would like to sort of welcome everybody to join us again next week when our speaker will be Sebastian Amigurena. And with this, I would like to thank everybody, wish everybody a great remaining day or a beautiful afternoon, and see you soon. Bye-bye.